So this is the outline of my presentation. For the exosome, I will not talk a lot because you already learned about the exosome in the previous lecture. As you already know that exosomes are the extracellular vesicle, which is used for the cell-to-cell communication, and they have different effect on your body, right? Okay, so what is the mechanisms of exosome in the management of the hair loss? So how exosome affect the hair loss? Actually, the mechanism is remain unclear about the use of exosome for hair loss. But it is suspected that exosome, through the modulation of the paracrine signaling, they can mediate the crosstalk between the epidermis cells and mesenchymal cells during the hair growth cycle. That means they can promote the hair growth, mediated by the epithelial cells and mesenchymal cells. So let's talk about this different source of the exosome. This is the adipose-derived stem cell exosome. Here you will see that this is the adipose-derived stem cell, and then this is the exosome. And if you have the cell culture with the adipose-derived stem cell exosome, you will have the lesser level of the MIR22, and then so if you have the lesser level of MIR22, there will be proliferation and apoptosis like this. Okay. Also, another mechanism is that exosome mediates to the WNT signaling pathway and increase beta catenin, and it can cause the hair growth induction. So another study in the mice, here you will see that so they can shift the telogen to the anagen by the exosome, and then so you will have the hair follicular regeneration. This is just the animal study, and this is just the cell culture study, in vitro study. Okay, but for this slide, in conclusion here, they can say that paracrine regulation of the dermopapillary cell and increase the hair growth and dermis thickness, but it's not tested in your body, it's tested in the laboratory and animals. Okay, the first thing. The second thing is about another source of the exosome is from your dermopapillary cell. This slide I talk about the adipose-derived stem cell exosome. This one is from your dermopapillary cell-derived exosome. They also have effect on the hair growth in mice and also in the lab. This is the mechanism that they promote the hair growth. Okay, they can induce the hair follicular growth, and they can accelerate the transition of the telogen to energy in mice, and then so they can also regulate the hair follicular stem cell, so that you can use that stem cell for the further growth of your hair. Okay, so this is about the dermopapillary cell. When we talk about the DPC exosome, we have to understand the microRNA. There are 111 different microRNA identified for the use of for the role in androgenetic alopecia. But here you will see that microRNA 22,5B inhibit the hair growth by downregulating like this. And another thing is that if you use the microRNA 181 and 72185P, this can promote the hair growth. So for this slide, please remember that for the dermopapillary cell-derived exosome, not all the cells can promote the hair growth. If you use only the cell with this microRNA, it can promote the hair growth. Otherwise, if you have this kind of microRNA, it can inhibit the hair growth as well. So what will be other? So the first thing is about adipose-derived stem cell. The second thing is about the dermopapillary. Another thing, these are the other source of exosome. From the outer root sheath stem cell, you can use outer root sheath cell. Also, you can also use the myeloid-derived suppressor cell, also the amniotic fluid cell, also the platelet PRP or something like that. However, there is an interesting result from this in vivo study here. If you extract the exosome from the platelet lysis or PRP, the effect is not that good in the laboratory. So this is the thing that we have to learn according to the research. So what about the clinical evidence? This is the research recently published in November related to the use of exosome in AGA. They did the research with 30 males, 22 and 65 years, AGA stage 3 and 6. And then they use the exosome from the foreskin-derived mesenchymal stromal. That means it is from the newborn prepuce. So prepuce of the newborn, they can generate the exosome. So they use that kind of exosome, foreskin-derived exosome, and they use 3 ml total by an patch technique, meaning the light injection to your scalp. And so they monitor the side effect 1 hour post-procedure, and they ask the patient to come at the 4 and 12 weeks of the post-injection. This is their research. So there is statistically improvement in the hair growth, mean density, hair density, at the 12 week and 4 week. Here, the statistically improvement, but there is no control group. They just do the open label study. There is no control group, and they just only monitor the side effect for just 1 hour and 4 week and 12 week. And then after that, the patient cannot come to their research center again, and so no safety data. So although they are affected, we cannot say that is this effective in long-term use or is it safe in long-term use? We cannot say like this. Okay, so we have to do effect or repeated exosome injection need to be evaluated. So this is their study finding here. After 4 weeks, you will see like this, and after 12 weeks, it will be like this. And then so exosome is good, but this is just a small study with only 30 patients, no control group, and they just test only for the 1 injection. And then after that, assessment in 4 weeks and 12 weeks, that's all. That's all. So we have to do more research. This is another study published in September 2024. This is related to adipose stem cell-derived exosome. Okay? They also use only 30 patients with male and female. They designed a six-month study. 6-month study. And then so they use this exosome mixed with the 2 mesaline, and then for this research, they use the microneedling for the application of the exosome. For the previous one, they used the injection, but here they use the microneedling for the exosome. And then so for the treatment schedule is like for the first treatment, they give the microneedling session for every 2 weeks for 7 sections. And then after that, next treatment, they give every 3 weeks for the next 3 sessions. So the patient usually receives 10 sessions of the exosome application. So what is their research? What is their results? So here you will see that at the 12 weeks and 24 weeks, hair density is significantly increased. And then here you can see that the density of the hair is much increased at the 24 week. For the advanced events, there are 5 participants. They complained the tingling after the microneedling session, but they said that it was tolerable, and then it resolved post-treatment, and then some erythema were noted by their physician, but they did not report any long-term result, long-term side effect. Okay, so interestingly, there are some research related to the use of exosome, clinical use of exosome by injection or by microneedling for AGA, but their studies are so small. So we need to do more about the safety and efficacy of the exosome. This is a study with the 39 patients, and then so they also found that the result is good. Mean hair density improved significantly, mean hair thickness improved significantly, also no side effects. Another study, they used the bone marrow mesenchymal cell-derived exosome, and then they measure the outcome for 6 months, and then so they also report that hair growth is seen in the 64.5%, and then so increase in the hair density was improved. But here, exosome impact is uncertain, and then so participants were allowed to use other treatment during this study. That means during their course of treatment, for some patients, they use the minoxidil, and then so they continue using the minoxidil. For some patients, they take the finasteride, so they continue taking the finasteride during their treatment session, and then so we are not sure about exosome have this effect or the other drugs have this kind of reversal of the AGA. So this is their weakness. Okay, how about the safety? So according to the previous paper that I mentioned earlier, for the topical use, no serious adverse event. For the injection, no reported adverse reaction, right? So for the topical use in Agnieszka, they said that the reported symptom will be the pain and erythema, but it will resolve spontaneously. So this is also the paper related to the safety study related to the exosome. Here, they conducted in vitro as well as in vivo, that means in the lab as well as in human. So this is their conclusion. So if they couldn't see any effect of the exosome on the liver, kidney, brain, and the lungs in the lab. Also here, there is no advanced evidence. If you use the exosome, you cannot say that you will have the hemolysis. No hemolysis, no DNA damage, no abnormal histological finding. So they said that there is no serious adverse events related to the use of exosome. However, in some other paper, here they mentioned that to swap in the cancer therapy, some exosome can promote the vascular tumorigenesis, angiogenesis, and metastasis. So some exosomes have different role. But for the other study, they have tumor suppressing effects. So we are not sure which exosome can promote the tumor, which exosome can be used for the tumor suppression. So that's why no exosome products are approved until now, as well as in the US, as well as in Thailand as well. They even give the warning. Here, these unapproved product, whether recover from your own body or another person's body, like stem cell, SVF, like this, and exosome. So they can cause blindness, tumor formation, infection, and more. And then so they said that these are unapproved. They give the warning like this. So we inject the exosome, we use the exosome in the microneedling, it is just off-label. But we have to think that there may be side effects like this, but we cannot say definitely, because there are still much research needed about this. Okay, so if you wish to do exosome in the future, there will be so much regulatory control. Okay, how about the future direction? So further optimization will be needed. If you use the exosome, they prefer to use the topical formulation rather than injection. It will probably be safer than the other. And also combination treatment. How about the combination treatment? Exosome combined with other therapy may increase the outcome. Here they combine with the minoxidil, they combine with the PRP, right? So there are also some novel drugs delivery methods. You can use the microneedling. Now, microneedling patch are very popular. You can put the exosome into this patch. And also, there are also studies related to the low-frequency ultrasound. You can use low-frequency ultrasound for delivery of the exosome. And so they said that human-derived exosome, even from your body or even from the other part of the body, is not good, right? So they are trying to make the exosome from the plants. This is the plant-based exosome, ashwagandha seeds. So they are trying to make like this. And then so for the take-home message, during these recent years, for around 10 years ago, stem cells are very popular. But they proved that stem cells, they have the effect on the tumorigenesis. And then so they try to extract the exosome because they are very tiny nanoparticle, which is used for the paracrine regulation each other, right? So they focus on the exosome research rapidly. And then so I just mentioned before, the latest research related to AGA. So many significant advances during this year. However, there are still challenges because their studies are very small. There is no control group. They only study the effectiveness and efficacy. They cannot mention about the long-term safety. So we are not sure about the long-term safety. Can they promote the tumorigenesis, or can they have other adverse effects? We are not sure about this. So we have to do more research. Safety issue is the key concern. So for the current exosome use is just off-label. Primary limit, so if you wish to use the exosome, they recommend only to use the topical exosome, and you can do the microneedling or apply it, do not inject this, just like this. So they are not sure about the safety of the exosome, and so for the future, they will do, they will do more research related to the exosome, and so we will probably prevent the effect of the exosome, not only from the AGA, but also from the other diseases in the skin or other part of the body. So this is the end of my presentation. Thank you so much for your kind attention.